Hi friends, my name is Nandita. I am from Andhra Pradesh.
Good morning dear students. Today we are going to discuss about plant growth and development from chapter 15 CBSE syllabus class 11. Children, you have already studied the organization of a flowering plant in chapter 5. Have you ever thought about where and how the structures like roots, stems, leaves, flowers, fruits and seeds arise, and that too in an orderly sequence? You are by now aware of the terms seed, seedling, plantlet, mature plant, etc. You have also seen that trees continue to increase in height or growth over a period of time. However, the leaves, flowers, and fruits of the same tree not only have limited dimensions but also appear and fall periodically and sometimes repeatedly. Why does vegetative phase precede flowering in a plant? All plant organs are made up of a variety of tissues. Is there any relationship between the structure of a cell, a tissue, an organ, and the function they perform? Can the structure and the function of this be altered? All cells of a plant are descendants of a zygote. The question is then why and how do they have different structural and functional attributes? Development is the sum of two processes, growth and differentiation. To begin with, it is essential and sufficient to know that the development of a mature plant from a zygote follow up a precise and highly ordered successions of events. During this process, a complex body organization is formed that produces roots, leaves, branches, flowers, fruits, and seeds, and eventually they die. The first step in the process of plant growth is seed germination. The seed germinates when favorable conditions for growth exist in the environment. In absence of such favorable conditions, the seed do not germinate and goes into a period of suspended growth or rest. Once favorable conditions returns, the seed resume metabolic activities and growth takes place. In this chapter, you shall also discuss some of the factors which govern and control this developmental process. These factors are both intrinsic means internal, and eccentric means external to the plant. Children, now let us discuss about growth. Growth is regarded as one of the most fundamental and conspicuous characteristics of a living being. What is growth actually? Growth can be defined as an irreversible permanent increase in size of an organ or its parts or even an individual cell. Generally, growth is accompanied by metabolic process, both anabolic and catabolic, that occurs at the expense of energy. Therefore, for example, expansion of a leaf is growth. How would you describe the swelling of a piece of wood when placed in water? Think about it. Children, now let us discuss about plant growth. Generally, it is intermediate plant growth is unique because plants retain the capacity of unlimited growth throughout their life. This ability of the plant is due to the presence of meristems at certain locations in their body. The cells of such meristems have the capacity to divide and self-perpetuate. The product, however, soon loses the capacity to divide, and such cells make up the plant body. These forms of growth, wherein new cells are always being added to the plant body by the activity of the meristem, is called the open form of growth. What would happen if the meristem ceases to divide? Does this ever happen? In chapter 6, you have studied about the root apical meristem and shoot apical meristem. You know what they are responsible for the primary growth of the plants and principally contribute to the elongation of the plant along their axis. You also know that in dicotyledonous plants and gymnosperms, the lateral meristem means vascular cambium and crop cambium appear later in life. These are the meristem that active. This is also known as secondary growth of a plant. Children, now let us see how growth is measurable. Growth at cellular level is principally a consequence of increase in the amount of protoplasm. Since increase in protoplasm is difficult to measure directly, one generally measures some quantity which is more or less proportional to it. Growth is therefore measured by a variety of parameters, some of which are increase in fresh weight, dry weight, length, area, volume and cell number. You may find it amazing to know that one single maize root apical meristem can give rise to more than 17,500 new cells per hour, whereas cells in a watermelon may increase in size up to 3,50,000 times. In the former, growth is expressed as increase in cell number, that later express growth as increase in size of the cell. While the growth of a pollen tube is measured in terms of its length, and increase in surface areas denotes the growth in an dorsiventral leaf. Children, now let us discuss about phases of growth. The period of growth is generally divided into three phases, namely meristematic, elongation and maturation. Let us understand this by looking at the root tips. The constantly dividing cells both at the root apex and the shoot apex represented the meristematic phase of the growth. The cells in this region are rich in protoplasm, possess large conspicuous nuclei. Their cell walls are primary in nature, thin and cellulosic with abundant plasmodesmatal connections. The cells proximal to the meristematic zone represent the phase of elongation. Increased vacuolation, cell enlargement and new cell wall deposition are the characteristics of the cells in this phase. Further away from the apex, that is more proximal to the phase of elongation, lies the proportion of axis which is undergoing the phase of maturation. The cells of this zone attain their maximal size in terms of wall thickening and protoplasmic modifications. Most of the tissues and cell types you have studied in chapter 6 represent this phase. Children, now let us discuss about growth rate. The increased growth per unit time is termed as growth rate. Thus, rate of growth can be expressed mathematically. An organism or a part of the organisms can produce more cells in a variety of ways. The growth rate shows an increase that may be arithmetic or geometrical. In arithmetic growth, following mitotic cell division, only one daughter cell continues to divide while the other differentiates and matures. The simplest expression of arithmetic growth is exemplified by a root elongation at the constant rate. On plotting the length of the organ against time, a linear curve is obtained. Mathematically, it is expressed as LT is equal to LO plus RT. LT means length at a time, T means time, LO means length at a time, 0 R is equal to growth rate and elongation per unit time. Let us now see what happens in geometrical growth. In most systems, the initial growth is slow and it increases rapidly thereafter at an exponential rate. Here, both the pregnancy cells following mitotic cell division retain the ability to divide and continue to do so. However, with limited nutrient supply, the growth shows down leading to a stationary phase. If we plot the parameter of growth against time, we get a typical sigmoid or S curve. A sigmoid curve is the characteristic of living organisms growing in a natural environment. It is typical for all the cells, tissues and organs of a plant. Can you think of more similar examples? What kind of curve can be expected in a tree showing seasonal activities? The exponential growth can be expressed as W1 is equal to WO. W1 means final size, means weight, height and number, etc. WO means initial size at the beginning of the periodic, R growth rate, T time of growth, E base of natural logarithms. Hence, R is the relative growth rate and is also the measure of the ability of the plant to produce new plant material, referred to as efficiency index. Hence, the final size of W1 depends on the initial size of WO. Quantitative comparisons between the growth of living system can also be made into two ways. One is measurement, and the comparison of total growth per unit time is called absolute growth rate. The growth of the given system per unit time expressed on a common basis, for example, per unit initial parameter, is called relative growth rate. In two leaves A and B are drawn that are of different sizes but shown absolutely increasing in the area in the given time to given leaves. A1 and AB, however, one of them shows much higher relative growth. Children, now let us discuss about the conditions for growth. Why do you not try to write down what do you think are necessary conditions for the growth? This list maybe have water, oxygen and nutrients has very essential elements for the growth. The plant cells grow in a size of cell enlargement which in turn require water. Turgidity of cells helps in extension growth. Those plant growth and further development is intimately linked to the water status of the plant. Water also provides the medium of enzymatic activities needed for growth. Oxygen helps in releasing metabolic energy essential for growth activities. Nutrients are required by plant for the synthesis of protoplasm and act as a source of energy. In addition, every plant organism has an optimum temperature range best suited for its growth. Any deviation from this range could be detrimental to its survival. Environmental signals such as light and gravity also affect certain stages of growth. Children, now let us discuss about differentiation, dedifferentiation and redifferentiation. The cells derived from the root apical and shoot apical meristem and cambium differentiate and mature to form specific function. This act lead to maturation is termed as differentiation. During differentiation, cells undergo few to major structural changes both in their cell walls and protoplasm. For example, to form a tertiary element, the cells would close their protoplasm. They also develop a very strong elastic lingocellulosic secondary cell walls to carry water to long distance even under extreme tensions. Try to correlate the various anatomical features you encountered in plants to the functions they perform. Plants show another interesting phenomena. The living differentiated cells that by now have lost the capacity to divide can regain the capacity of division under certain conditions. This phenomena is termed as dedifferentiation. For example, formation of meristem, intravascular cambium and cork cambium from fully differentiated parenchyma cells. While doing so, such meristem tissues are able to divide and produce cells that once again lose the capacity to divide but mature to perform specific functions, that is, get redifferentiated. List some of these tissues in a woody dicotyledonous plants that are products of redifferentiation. How would you describe a tumor? What would you call the parenchyma cells that are made to divide under controlled laboratory conditions during the plant tissue culture? We have mentioned that the growth in plants is open, that is, it can be intermediate or determinate. How we may say the event differentiation in plants is open because the cells and tissues arising out of the same meristem have different structures at maturity. The final structure at the maturity of a cell or a tissue is also determined by the location of the cell within. For example, cells positioned away from the root apical meristem differentiate as a two root cap cells, while those pushed to the periphery matured as epidermis. You can add a few more examples of open differentiation correlating the position of a cell to its position in an organ. Children, now let us discuss about development. Development is a term that includes all the changes that an organism goes through during its life cycle from germination of a seed to senescence. Diagrammatic representation of the sequence of process which constitute the development of a cell of higher plants is given in the figure. It is also applicable to tissues and organs. Plants follow different pathways in response to environment or phases of life to form different kinds of structures. This ability is called plasticity. Heterophyly in cotton, coriander, and larkspur. In such plants, the leaves of the jejunal plant are different in shape from those in mature plants. On the other hand, difference in shapes of leaves produced in the air and those produced in water in buttercup also represent the heterophylosis development due to the environment. This phenomena of heterophyly is an example of plasticity. Children, now let us discuss about plant growth regulation. Characteristics the plant growth regulations are small, simple molecules of diverse chemical positions. They would be indole compounds, adenine derivatives, derivatives of carotenoids, terpenes acid, gaseous, etc. Plant growth regulates are variously described as plant growth substance, plant hormones or phytohormones in literature. The PGR can be broadly divided into two groups based on their functions in a living plant body. One group of PGR are involved in growth promoting activities such as cell division, cell arrangement, pattern formation, trophic growth, flowering, fruiting, and seed formation. There are also called plant growth promoters, for example, auxins, gibberellins, and cytokinesis. The PGR of the other group play an important role in plant response to wounds and stresses of biotic and abiotic origin. They are also involved in various growth inhibiting activities such as dormancy and abscissance. The PGR abscisse acid belongs to this group. The gaseous PGR ethyl could fit either of this group, but it is largely an inhibitor of growth activities. Children, now let us discuss about discovery of plant growth. The plant growth regulators, interestingly, the discovery of each of the five major groups of PGRs have been accidental. All this started with the observations of Charles Darwin and his son Franz Darwin when they observed that the scolepitils of canary grass respond to unilateral illuminations by growing towards a light source, which is called phototropism. After a series of experiments, it was concluded that the tip of coleoptile was the site of transmittable influence that caused by bending of entire coleoptile. Auxin was isolated by FW went from the tips of coleoptiles of occult seedling. The Bakane means foolish seedling disease of rice seedling was caused by a fungal pathogen Gibberella was what caused by a fungal pathogen Gibberella. E. Kurosawa in 1926 reported the appearance of symptoms of these diseases in rice seedling when they were treated with sterile filters of the fungus. The active substance were later identified by gibberellic acid. Skoog and his co-workers observed that the form of internodal segments of tobacco stems, the callus proliferated only if, in addition to auxins, the nutrients medium was supplemented with one of the followings: extract of vascular tissues, yeast extract, coconut milk or DNA. Miller et al. Later identified and crystallized the cytokinesis promoting active substance that they termed chitin. During mid 1963, independent researchers reported the purification and chemical characterization of three different kinds of inhibitors: inhibitor B, abscissans 2, and dormin. Later, all these three were proved to be chemically identical. It was named as Asbestic acid ABA-HH fusions in 1910 confirmed the release of volatile substance from ripened organs that hastened the ripening of stored unripe bananas. Later, this volatile substance was identified ethyl gaseous PGA. Let us study some of the physiological effect of these 5 categories of PGA in next section. Children, now let us discuss about physiological effects of a plant growth regulators. Auxins was the first. It is isolated from human urine. The term auxin is applied to the iodine 3 acetic acid and to other natural and synthetic compounds having certain growth regulating properties. They are generally produced by growing apices of the stem and roots, from where they are migrate to the region of their action. Auxins like IAA and inodyl butyric acid have been isolated from plants. NAA and 2,4-D as synthetic auxins. All these auxins have been used extensively in agricultural and horticultural practices. They help to initiate rooting in stem cutting and application widely used for the plant propagations. Auxins promote flowering. Example, pineapples. They help to prevent fruit and leaf drop at earlier stages but promote the ascension of older mature leaves and fruit. In most higher plants, the growing apical bud inhibits the growth of lateral axillary buds, a phenomena called apical dominance. Removal of a shoot tip usually results in the growth of lateral buds. It is widely applied in tea plantations, hedge making, etc. Can you explain why auxins also induce parthenography, that is in tomatoes? They are widely used as herbicides, means 2,4-D widely used to kill dicotyledonous weeds does not affect mature monocotyledonous plants. It is used to prepare weed fire lawns by gardens. Auxin also controls xylem differentiation and helps in cell division. Children, now let us discuss about gibberellins. Gibberellins are another kind of promotory PGR. There are more than 100 gibberellins reported from widely different organisms such as fungi and higher plants. They are denoted as GA1, GA2, GA3 and so on. However, gibberellic acid GA3 was one of the first gibberellins to be discovered and remains the most intensively studied form. All GAs are acidic. They produced a wide range of physiological response in plants. Their ability to cause an increase in length of axis is used to increase the length of grape stalks. Gibberellins causes fruits like apple to elongate and improve its shape. They also delay senescence. Thus, the fruit can be left on the tree longer so as to extend the market period. GA3 is used to speed up the malting process in brewing industry. Sugarcane stores carbohydrate as sugar in their stems. Spraying sugarcane crop with gibberellins increase the length of the stem, thus increasing the field by as much as 20 tons per acre. Spraying juvenile coniferous with GAS hastens maturity period, those leading to earlier seed production. Gibberellins also promotes bolting, internodes elongation, just flowering in beet, cabbages and many plants with the roast habit. Children, now let us discuss about cytokinesis. Cytokinesis have specific effects on cytokinesis and were discovered by kinetin from the autocalved hearing sperm DNA. Kinetin does not occur naturally in plants. Search for natural substance with kinetin like activities leads to isolation of gelatin from corn kernel and coconut milk. Since the discovery of gelatin, several naturally occurring cytokinesis and some synthetic compounds with cell division promoting activity have been identified. Natural cytokinesis are synthesized in regions where rapid cell division occurs, for example, root apex, developing shoot buds, young fruits, etc. It helps to produce new leaves, chloroplast in leaves, lateral shoot growth, and adventitious shoot formation. Cytokinins help overcome the apical dominance. They promote nutrient metabolization, which helps in the delay of leaf senescence. Children, let us discuss about ethylene is a simple gaseous PGR. It is synthesized in large amount of tissues undergoing senescence and ripening fruits. Influence of ethylene on plants including horizontal growth of seedling, swelling of the axis and apical hook formation in dicot seedling. Ethylene promotes senescence and abscissence of a plant organ, especially of leaves and flowers. Ethylene is highly effective in fruit ripening. It enhances the respiration rate during ripening of the fruit. This rise in the rate of respiration is called respiratory climatic. Ethylene breaks seed and bud dormancy, initiates germination in peanut seeds, sprouting of potato tubes. Ethylene promotes rapid internodes, petiole elongation in deep water rice plants. It helps leaves upper parts of the shoot to remove above water. Ethylene also promotes root growth and root hair formation, thus helping the plants to increase their absorption surface. Ethylene is used to initiate flowering and for synchronizing fruit set in pineapples. It also includes flowering in mangoes. Since ethylene regulates so many physiological processes, it is one of the most widely used PGR in agriculture. The most widely used compound as a source of ethylene is ethophyne. The ethophyne in an aqueous solution is readily absorbed and transported within the plants and releases ethylene slowly. The ethophyne hastens fruit ripening in tomatoes and apples and accelerates abscissions in flowering and fruits. It promotes female flower in cucumber, thereby increasing the yield. Children, now let us discuss about abscisic acid. As mentioned earlier, abscisic acid ABA was discovered for its role in regulating abscissions and dormancy. But like other PGR, it also has other wide-ranging effects on plant growth and development. It acts as a general plant growth inhibitor and an inhibitor of a plant metabolism. ABA inhibits seed germination. ABA stimulates the closure of stomata and increases the tolerance of plants of various kinds of stresses. Therefore, it is also called the stress hormone. ABA plays an important role in seed development, maturation, and dormancy. By inducing dormancy, ABA helps to withstand desiccation and other factors unfavorable for the growth. In most situations, ABA acts as an antagonist to GAS. We may summarize that for any and every phase of growth, differentiation, and development of plants, one or the other PGA has some role to play. Such roles could be complementary and antagonistic. This could be individualistic and synergistic. Similarly, there are a number of events in the life of a plant where more than one PGR interact or to affect that event. Example, dormancy in seeds, buds, abscissions, senescence, apical dormancy, etc. Remember the role of PGR if of one kind of instructed control, along with the geomic control and extrinsic factor, they play an important role in plant growth and development. Many of the extrinsic factors such as temperature, light, plant growth and development via PGR. Some of such events could be vernalization, flowering, dormancy, seed germination, plant movements, etc. We shall discuss briefly the role of light and temperature, both of them the extrinsic factor or initiation of flowering. Children, now let us discuss about photoperiodism. It has been observed that some plants require a periodic exposure to light to induce flowering. It also seen that such plants are able to measure the duration of exposure to light. For example, some plants require the exposure to light for a period exceeding well defined critical duration, while the others must be exposed to light for a period less than this critical duration before the flowering is initiated in them. The former group of plants are called long day plants, while the later ones are termed as shorter day plants. The critical duration is differentiated for different plants. There are many plants, however, where there is no such correlation between exposure to light duration and induction of flowering response. Such plants are called day neutral plants. That not only the duration of light period but that the duration of dark period is also equal importance. Hence it can be said the flowering in certain plants depends not only on combination of light and dark exposures but also their relative durations. This response of plants to the periods of day and night is termed as photoperiodism. It is also interesting to note that while shoot epics modify themselves into flowering epicycles prior to flowering, they by themselves cannot perceive photoperiods. The site of perception of light and dark durations are the leaves. It has been hypothesized that there is a hormonal substance that is responsible for flowering. This hormonal substance migrates from leaves to shoot apex for inducing flowering only when the plants are exposed to the necessary inductive photoperiod. Children, now let us discuss about vernalization. There are plants for which flowering is either quantitatively or qualitatively depend on exposure to lower temperature. This phenomena is termed as vernalization. It prevents precocious reproduction development late in the growing season and enables the plant to have sufficient time to reach maturity. Vernalization refers specially to the promotion of flowering by a period of low temperature. Some important food plants, wheat, barley, rye, have two kinds of varieties, winter and spring varieties. The spring variety are normally planted in the spring and comes to flower and produce grain before the end of the growing season. Winter varieties, however, if planted in spring, would normally fail to flower or produce mature grain within a span of a flowering season. Hence, they are planted in autumn. The germinate and overwinter come out as small seedling, resume growth in spring, and are harvested usually around midsummer. Another example of vernalization is seen in a biennial plants. Biennials are monocarpic plants that normally flowers and die in the second season. Sugar beet, cabbage, carrots are some of them common by subjecting the growing of a biennial plant to the cold treatment stimulates a subsequent photoperiodic flowering response. Children, let us discuss about seed dormancy. There are certain seeds which fail to germinate even the external conditions are favorable. Such seeds are understood to be undergoing a period of dormancy which is controlled not by external environment but are under endogenous control or conditions within the seed itself. Impermeable and hard seed coat, presence of chemical inhibitors such as ascorbic acids, phenolic acids, ascorbic acid and immature embryos are some of the reasons which causes seed dormancy. This dormancy, however, can be overcome through a natural means and various other man-made measures. For example, the seed coat barrier in some seeds can be broken by a chemical abrasions using knives, sandpaper, or vigorous shaking, etc. In mature, these aberrations are caused by microbial actions and phases through digestive tract of animals. Effect of inhibitory substance can be removed by subjecting the seeds of chilling conditions or by application of certain chemicals like gibberellic acid and nitrates. Changing the environment conditions such as light and temperature are other methods to overcome seed dormancy.